Hey everyone, today we'll learn about glycogen, glycogen metabolism,
and gluconeogenesis. So to start with the basics, we need to review glycogen. Glycogen is a polysaccharide that serves as the primary short-term energy storage form of glucose in animal tissues. So basically, glycogen is the mobilized storage form of glucose. And glycogen breakdown, which we will talk about in later slides, provides substrates that enter glycolysis. So where is glycogen found? We know it's in animals, primarily the liver and muscles are the main storage sites. We know it is not in plants because plants use starch as their main storage carbohydrate instead of glycogen. Glycogen is also found in bacteria because, like animals, bacteria uses glycogen as short-term stored fuel sources. Okay, so moving on, we're going to now look at glycogen structure. So as you can see here, glycogen is highly branched chains of linked glucoses with many non-reducing ends. So continuing on with structure, we're going to look at amylopectin on the left, which is a starch, and glycogen on the right. Now, by just this picture alone, which one is more branched? As we can see, amylopectin, the starch, is less branched. Now, why is this? Because plants prioritize stable, dense, long-term storage, while glycogen is more branched because animals prioritize fast glucose availability. So let's take a closer look with this diagram I made of glycogen structure. So as you can see, the light blue dots are the non-reducing ends, and there are multiple of those on a glycogen structure. The dark blue or dark purple are the branch points, which there's also many of them throughout glycogen structures, but there's only one reducing end at the bottom right. Green is the one reducing end, and we'll talk about why that's important. So glycogen breakdown starts from the non-reducing ends, and glycogen only has one reducing end. And attached to this one reducing end is a protein named glycogen. So just to review before we move on, glycogen is a readily mobilized storage form of glucose, and glycogen breakdown supplies glucose for glycolysis. So now we're ready to move on to glycogen breakdown, or glycogenolysis. So in this glycogen breakdown, the major enzyme used here is glycogen phosphorylase. So glycogen phosphorylase is going to work on the non-reducing ends of glycogen, and basically is going to break the 1-4 linkage and release a glucose 1P. And just to note here, there are other enzymes used in moving glycosyl units and breaking branching points and releasing free glucose. But this is the major one used. So let's take a deeper dive into this major enzyme. Glycogen phosphorylase cleaves by addition of orthophosphate to yield glucose 1-phosphate. So if you can see the arrow pointing to the top right diagram, you have a glycogen. Glycogen phosphorylase is cleaving by the addition of orthophosphate, and then the product is glucose 1-phosphate. Phosphorylysis is the cleavage of a bond by the addition of orthophosphate. So this is what's happening in the top right reaction. And what the glycogen phosphorylase is doing is catalyzing the sequential removal of glucosyl residues. To turn our attention to the bottom right diagram, what's being shown here is orthophosphate splits the glycosidic linkage between the carbon 1 of the terminal residue, which is the non-reducing end, and the carbon 4 of the adjacent one. And that is all circled in red right over there. Let's look at how glucose 1-phosphate is converted to glucose 6-phosphate. Basically, glucose 1-phosphate is released from glycogen and is then converted to glucose 6-phosphate by the enzyme phosphoglucomutase. This reaction takes no ATP. And the enzyme phosphoglucomutase shifts the phosphate within the same molecule. In this, one ATP is saved for glycolysis, except the glucose released by alpha-1,6 glucosidase from the branching point. Glucose from branch points is released as a free glucose, so it requires one ATP. This is important because G1P, glucose 1-phosphate, can't directly enter glycolysis, but G6P, glucose 6-phosphate, can. When the muscle gets this glucose 6-phosphate, it goes straight into glycolysis, while when the liver gets it, it converts it to glucose. and then the glucose is released into the blood. A major function of the liver is to maintain a constant blood glucose level. During physical activity and between meals, the liver supplies glucose to the blood, which is then used by the brain and skeletal muscle. Glucose with a phosphate group cannot be transported out of the cell and must be dephosphorylated first. This is shown in the reaction below. When you have a glucose 6-phosphate plus H2O, water, turned into glucose and an inorganic phosphate. The catalytic enzyme for this reaction is glucose 6-phosphatase. Now, to break glycogen down, it needs to debranch. In the first step of debranching of glycogen, phosphorylase breaks the alpha-1,4 carbon bonds. Glycogen phosphorylase removes glucose residues from the non-reducing ends, releasing glucose 1-phosphate until it reaches a branch point, then moves to the second step, the transfer step. The debranching enzyme moves a block of 3 glucose residues from the short branch to another nearby chain, leaving only 1 glucose at the branch point, which then moves on to the cleavage step. The debranching enzyme hydrolyzes the remaining alpha-1,6 bond, releasing the last glucose as free glucose. As a result, the branch point is removed, allowing glycogen phosphorylase to continue breaking the chain completely. Now that we talked about the breaking down of glycogen, we can talk about glycogen synthesis, or glycogenesis. The major enzyme used in glycogenesis is glycogen synthase. This enzyme transfers the glucose from UDP glucose to a non-reducing end of glycogen, making an alpha-1,4 glycosidic bond. Glycogen synthase then extends the chain at the non-reducing ends via alpha-1,4 linkages, which I just talked about, while a transferase enzyme forms branches. Now, what is UDP? UDP glucose is the glucose donor. So, as you can see the formula of the reaction above, glycogen N plus UDP glucose yields glycogen N plus 1 plus UDP. So, for whatever amount of glycogen you put in, plus 1. So if I put in 3 glycogen, it'll yield 3 plus 1 is 4 glycogen. So more on this UDP glucose, or uridine disphosphate glucose. So UDP glucose is the glucose donor in the biosynthesis of glycogen that donates glucose units during glycogen synthesis. The C1, carbon 1 of glucose, in UDP is activated because its hydroxyl group is linked to a UDP via a disphosphate bond. But how is this formed? Basically, when glucose 6-phosphate builds up, it is converted into glucose 1-phosphate to begin glycogen synthesis. UDP glucose is formed from glucose 1-phosphate and UTP by the enzyme UDP glucose pyrophosphorylase. During this reaction, the outer 2 phosphates of UTP are released as pyrophosphate, and this can all be shown in the diagram above. So now this UDP glucose is used in glucose transfer to glycogen because it donates a glucose unit to the C4 hydroxyl of the chain end, creating an alpha-1,4 glycosidic linkage. The terminal hydroxyl group of the growing glycogen chain displaces UDP. And then a new glucose unit is added to the non-reducing end of the chain. And this can all be seen on the left. So I know all I've been talking about is a lot. So just to summarize, glycogen is the storage form of glucose in animals and is broken down to release glucose when energy demand is high. When the body doesn't need energy, glucose is stored as glycogen. Glucose is incorporated into glycogen using UDP glucose as the intermediate, which we just talked a lot about. So get used to all this material before we start talking about glycolysis versus gluconeogenesis. So what is gluconeogenesis? It's the synthesis of glucose from small non-carbohydrate carbon substrates such as pyruvate, lactate, glycerol, glucogenic amino acids, and this occurs mainly in the liver. Well, how is this all different from glycolysis? Well, glycolysis is catabolic. It starts with a 6-carbon molecule, and is then split up into multiple 3-carbon molecules. It is also oxidative. Its NADH is reduced. It yields energy. Its net yield is 2 ATP, and the reaction has a negative delta G, so it is spontaneous and exergonic. This completely differs from gluconeogenesis because it's anabolic. Its smaller molecules are upgraded to a 6-carbon molecule. It's reductive. The NADH is oxidized instead of reduced. It actually requires energy. It costs ATP and GTP. And its delta G is positive, so it's non-spontaneous and an endergonic reaction. I don't want you to confuse gluconeogenesis with glycogenesis. Glycogenesis is the converting of glucose into glycogen for storage, whereas gluconeogenesis is making new glucose from non-carbohydrate sources like lactate, amino acids, and glycerol. I just want to emphasize this one more time. So glycolysis only has 3 irreversible reactions. Gluconeogenesis does not use the same enzymes in these reactions, and 7 reactions are reversible, and this is where gluconeogenesis uses the same enzymes. Let's look at these 3 irreversible glycolysis reactions. The first reaction uses pyruvate kinase. In gluconeogenesis, instead of pyruvate kinase, it replaces it with 2 reactions requiring 2 ATP or 2 GTP, since 2 pyruvate equals 2 PEP. The second reaction uses PFK1. Gluconeogenesis replaces PFK1 with a phosphatase enzyme. The third reaction uses hexokinase. Gluconeogenesis replaces hexokinase with a phosphatase. And these are all the workarounds, quote-unquote workarounds, that gluconeogenesis has for these irreversible glycolysis reactions. The first step from gluconeogenesis from pyruvate is carboxylated to oxalaloacetate, which requires biotin, a CO2 carrier. Pyruvate carboxylase is subject to allosteric control. It is activated by acetyl-CoA, and this costs 1 ATP. As you can see in the diagram above, oxaloacetate is an intermediate. Next, this intermediate undergoes decarboxylation coupled with GDP-dependent phosphorylation to produce PEP. This net reaction of carboxylation and decarboxylation is pyruvate plus ATP plus GDP yields phosphenolpyruvate plus ADP plus GDP plus PI. So now let's look at this diagram of the first step. Pyruvate is converted to oxaloacetate in the mitochondrial matrix by carboxylation using ATP. As you can see, pyruvate goes in to the mitochondria. In the mitochondrial matrix, oxaloacetate is converted to malate through NADH-dependent reduction. Malate is then transported to the cytoplasm, where it is oxidized to oxaloacetate, generating NADH and then converted to PEP. Basically, pyruvate comes in, malate comes out. Once oxaloacetate is formed, it is converted to PEP, and gluconeogenesis proceeds through several sugar phosphate steps, which we will now go over. The reaction shown above is the hydrolysis of glucose 6-phosphate to glucose and 1 inorganic phosphate. This reaction is spontaneous and catalyzed by glucose 6-phosphatase, not hexokinase, which we talked about in previous slides, which is very important. One other different reactions in gluconeogenesis relative to glycolysis involve phosphate ester bonds bound to sugar hydroxyl groups being hydrolyzed. In this reaction, we have fructose 1,6-bisphosphate instead of phosphofructose kinase, or PFK1. An allosteric enzyme inhibited by AMP and activated by ATP. This is just the reverse of PFK1, which is very important and crucial to remember. There are 4 important substrates for gluconeogenesis. The first is pyruvate, which we've been talking about. The second is lactate. Lactate is recycled into glucose by the liver. The third is glycerol. Glycerol from fat breakdown is converted into glucose in the liver via DHAP. The fourth important substrate of of gluconeogenesis is alanine. Alanine carries carbon from muscle to the liver where it is used to make glucose. Let's start by looking at the second substrate we just talked about, lactate. So, the Cori cycle is where the liver remanufactures glucose from lactate for tissue use. During intense anaerobic exercise, muscle glycolysis converts glucose to pyruvate, and NADH+ is regenerated by reducing pyruvate to lactate. Lactate then goes to the liver, becomes pyruvate, and is used to make glucose. Then the liver sends glucose back to the muscle for glycolysis. Thus, the liver helps support muscle during intense exercise. Skeletal muscle primarily carries out glycolysis, while the liver primarily performs gluconeogenesis. The third substrate for gluconeogenesis is glycerol. Glycerol production is the hydrolysis of triglycerol in adipose tissue, or fat cells. Glycerol is transported via the blood from adipose tissue, the fat cells, to the liver, where it is taken up and converted to DHAP. DHAP combines with glyceraldehyde 3-phosphate via aldolase to form fructose 1,6-bisphosphate, which is then converted to glucose through gluconeogenesis. The last and final substrate for gluconeogenesis is alanine. Alanine carries carbon from muscle to the liver, where it is used to make glucose. Alanine is a major precursor for gluconeogenesis. It is formed in muscle from pyruvate via transmission, transported through the blood to the liver. Alanine turns into pyruvate, and then pyruvate enters gluconeogenesis, which creates glucose. So 2 very important things to remember as this video is coming to an end. The major site of gluconeogenesis is the liver, and glucose can be synthesized from non-carbohydrate precursors. The final thing I'll talk about in this video is glycolysis and gluconeogenesis' regulation. So I made this little graphic here. So basically, glycolysis and gluconeogenesis are regulated in opposite directions to maintain blood glucose and energy balance. Glycolysis is stimulated when glucose is abundant and energy is needed. Key enzymes such as PFK1 and pyruvate kinase are activated by AMP and insulin, while ATP and citrate inhibit the pathway. Gluconeogenesis is activated during fasting or low blood glucose to produce new glucose, mainly in the liver. Enzymes such as fructose 1,6-bisphosphatase are stimulated by glucagon and high ATP, but inhibited by AMP and insulin. This reciprocal prevents both pathways from being highly active at the same time. So basically, to completely simplify exactly what I just said, I made this little graphic here. So when ATP is high, it makes glucose. When ATP is low, it uses glucose. ATP acts as an energy sensor that basically balances glycolysis and gluconeogenesis. And that's all I have after regulation. So thank you for watching. I hope you learned something.